have, it's either the garden pea is wrinkled or smooth. So you can't measure that. You can't measure the level of smoothness or wrinkledness. So we say they are qualitative variation, physiological variation. Physiological variations are differences in the ways members of the same species behave or react to conditions in their environment. The way members of the same species behave or react to conditions in the environment. An example of physiological variation, we have ability to roll tongue. Some people can roll their tongue, while some cannot. Then the next one is the ability to detect taste of certain chemical substances. For instance, we have the phenylethylcarbamide, the PTC. Some people have that ability while others do not have it. So it's a yes or no thing. It's not that the person is averagely tall or so tall or whatever. You can't measure it. It's either a yes or a no. Then another physiological variation is high fidomcity, the ability to lay many eggs or produce more seed. That's what is called high fidomcity. Then another physiological variation is the blood groups. We have 4 major types of blood group. We have the blood group A, we have the blood group B, some people have the blood group AB, and others have blood group O. I'll be talking more about those 4 groups, but that's also another physiological variation. Then the next is fingerprints. Fingerprints, we also have 4 types. The kind of arrangement of ridges on your finger, especially your thumb, is what is called the fingerprint. And we have 3 types of fingerprints. We have the wall or compound fingerprints. The ridges form at least one complete circle. For people that have it, when you add a color, for instance, maybe a blue or whatever color or dye to your finger, and you place your finger, that the colored part, placing it on a white paper, you will see the type of fingerprints that you have. So there are some people having wall or compound fingerprint, some others have the loop, and others have the arc fingerprint. So when the ridges, that is the pattern of the fingers, that they form at least one complete circle, that is called wall or compound. Then we have the next one called loop. In this case, the ridges turn backward and run almost parallel to the ridges. So we call that the loop variation or the loop fingerprint. The third one is called the arc. And in this case, the ridges run in one general direction alone. Just in one direction alone, that is called the arc. Another type of physiological variation is sex, whether male or female. That's a physiological variation. Then variation in flowers, in the fruits, the shape, the color of seed, the leaf size, the root size, among plants of the same species. Now, what are the applications of variation? What importance they have, and how can we apply them to our day-to-day activity? First, variation can be employed in crime detection. There are no 2 people that have the same fingerprints. No. Even if you are twins, your fingerprints are different. So there are no 2 people that have the same fingerprint. Since that is the case, then criminals can easily be identified. For instance, the last person that got to a particular scene, maybe a scene of accident or a scene of gunshot, or they found the gun. They found a gun that was used to kill somebody. The technology can help us get the fingerprints of the person that held that gun. So the person can be traced, because no 2 people have that. But how the person can be traced is that in developed countries, everything has been documented. So once your name appears, every detail about you is there. So your fingerprint is seen, is known. So it can be identified easily. Then blood group of stains found at a scene of crime. For instance, maybe there was a fight or there was an argument that led to bloodshed. So the blood, the stains that was found at the scene can be taken to the lab for analysis, and the blood group of the person the blood group of the blood stain that was found at the scene of the crime can provide a clue to identifying the arrested suspects. You know, when an incident happens, you just gather people and call them suspects and start investigation. So when stains of blood are taken and blood group is taken, comparing it with the blood group of the suspects, they can be able to say, okay, truly it was there. Oh, this person was not at that scene. So variation is very, very useful in crime detection. Also, variation is useful in blood transfusion. Since everybody does not have the same type of blood, we have people with blood group A, some others with B, others with AB, and the fourth group we have the O. So, and the type of antigen that is present in the blood determines the blood group. So people with blood group A have antigen A in their blood. People with blood group B have antigen B in their blood. People with blood group AB have antigens A and antigens B in their blood group. So this is a table that just summarizes that. We have the blood group A, it has antigen A, it has antibodies, that's the agglutinin B. Then blood group A, people with blood group A can donate their blood to other people that has blood group A and people with blood group AB. So they can only donate to two groups, their own blood group and blood group AB. Then they can receive blood from blood group O and blood group A. So they can give, blood group A can give blood to their own type and can receive also from A. But they can also donate to blood group AB and receive blood from blood group O. The reason they can receive from blood group O is that there is no antigen in blood group O. So there will be no clamp or agglutination that will occur when they receive such blood. So we say that blood group O are universal donors. They can donate to anybody because there is no antigen in their blood. So there will be no agglutination when they donate. There will be no reaction. The antigens will not react. So that is why O is universal donor, while AB is universal recipient. You know, people with blood group AB have the antigen A and the antigen B in their blood. So they can receive from everybody. They can receive from A, they can receive from B, they can receive from AB, they can also receive from O, since O does not even have any in their blood. So blood group B they have antigen B and they have the agglutinin A. That's the opposite now. They have antigen B, but they have agglutinin A. That's anti-A. Then blood group B can donate to people with blood group B and also people with blood group AB. Then they can receive, blood group B can receive from blood group B and also blood group O. I said O, they are universal donors. Then for blood group AB has antigen A and antigen B. Then they don't have any antibody in their plasma. Then they can donate. They can only donate. Blood group AB, now note this. Blood group AB can only donate to blood group AB. They cannot donate to any other person. Then they can receive from everybody. See them. They cannot donate to any other person except people with their own blood group. But they can receive from anybody. They can receive from A, they can receive from B, they can receive from O, they can receive from AB. So they are universal recipients, while O is universal donor. Then for blood group O, they have no antigen in their blood, but they have antibodies, antibodies A and B. They have no antigen, but they have antibodies A and B in their blood. They can donate to everybody. They are universal donor. They can donate to O, they can donate to AB, they can donate to A, they can donate to B. But they can only receive from O. See the irony. AB can receive from everybody and can only donate to AB. O can give to everybody, but they can only receive from O. Now, the next application of variation, we talk about resource factor. Resource factors are certain substances called antigen, and they are called Rh factor. They occur in the red blood cells of a high proportion of human beings. So, a greater proportion of human beings have the Rh factor in their blood. but very few. Some people don't have that Rh factor in their blood. So the people that have the Rh factor in their blood are said to be Rh positive, while the people that don't have it are said to be Rh negative. For instance, when you do your blood test for blood group, they will tell you the result, whether you are blood group AB or O. But aside that, they will now put something like either a plus or a negative. So they can say you are O positive, O negative, B positive, B negative, A positive, A negative. So that positive or negative is talking about the rhesus factor. So we'll be looking at the importance of the rhesus factor in human beings. When a Rh negative, that is a rhesus factor, somebody that does not have that rhesus factor in his blood, when he receives blood from somebody that has it. So let's say, since I said O, the people with blood group O can only receive blood from group O, but it's also with a clause now. If the person is O positive or O negative, let's say the person is O negative and he receives blood from O positive, there will be a reaction in his blood, and his blood will produce toxic substances and antibodies that may cause clumping. So the rhesus factor is also very important in blood donation. So they will ask whether you are positive or negative to the Rh factor. When an Rh negative pregnant woman carries a baby with Rh positive pregnant fetus, that is, the mother is Rh negative, but the baby now, the fetus in her womb, is Rh positive, her blood may manufacture anti-Rh negative antibodies. You know, since she has Rh, her blood will manufacture something that will counter the Rh factor of the baby in her womb. So it might diffuse back into the circulatory system of the baby through the placenta, whereby there will be a reaction, and the Rh positive antigen of the fetus red blood cells. This will result in the red blood cells of the baby being destroyed, especially in the second pregnancy. The body might still tolerate it for the first pregnancy, and that is why people that do abortion, sometimes they don't know the implications of what they do, because sometimes they don't even know if they are positive or negative. They don't even know about whether there is any Rh factor or not. So if somebody